I'm Michael Freeman. I'm a current second-year master's student at the University of Washington in Patrick Tobin's Disturbance Ecology Lab, and I'll be presenting my research on managing for ecosystem services and the role of biotic agents in Douglas fir decline in the Western Cascades. So my research has two different sides to it, both of which have to do with Douglas fir mortality, one of which is more focused on bark beetles as a vector for that. So I'm going to focus first on the first part of my project on Douglas fir bark beetles, and then I'll get to the actual mortality later on. So for this first objective, I'm looking to quantify Douglas fir beetle adult phenology and seasonality and determine the primary temperature drivers of adult beetle flight. And temperature is actually the primary driver of insect activity, so determining that relationship for each species is crucial for understanding the seasonality of that species. So I'll give some quick background on the Douglas fir bark beetle. It's a native insect. It's native to here. It's a normal part of mortality in succession in our regional forests in the Pacific Northwest. Of course, as its name suggests, its host is the Douglas fir, Pseudosugum incesii, and it is believed to be a obligate univoltine insect. And so since it's a bark beetle, it reproduces underneath the bark of Douglas fir. So that begins when an adult female burrows through the bark of a Douglas fir. Usually more mature trees are preferred. And once it burrows in, it reaches the living phloem. And then once she reaches the phloem, she'll burrow vertically up the tree. Now she's burrowing vertically. Once she's mated with a male, she's laying eggs. And this is the creation of what we call a gallery. That's the picture in the upper left, is what this will eventually look like. And once those eggs hatch, the larvae will eat their way around the tree through that very dense, high-energy phloem, like in that picture on the top left, and they're consuming and they're going through the different instars of growth, and eventually they'll pupate and become adults, and then they'll emerge as adults and fly around, and the process repeats. And so these insects aren't necessarily the cause of mortality, but they are an agent of mortality. It's kind of like how when a human gets AIDS, it's not necessarily the AIDS that kills the person, it's the common cold. Douglas fir bark beetle works in a similar way. So when a tree is already stressed by some sort of other physiological stressor, that's when the Douglas fir bark beetles come in and kind of finish the job and kill the tree. But they're a very good way to measure overall stand resilience and health as kind of an easier to measure variable. And so these, as Douglas fir bark beetles, they can only affect living trees. They will not burrow into dead trees. That's only specific to this species. Some other species of bark beetles can go into dead trees, but this species will only affect living trees. And as I said, it's believed to be an obligate univoltinism. That means that it's believed that this species needs to and can only reproduce once per year. However, some new research suggests that it may in fact be facultative univoltinism, including my own study, which I'll get to. But that means that it's possible the only reason they reproduce once per year is just due to temperature conditions. So I have a little representation of how this process works. So this represents a healthy, not stressed Douglas fir stand. So in a healthy Douglas fir, they have an active immune system as it relates to Douglas fir bark beetles. And the way that it works is when a Douglas fir bark beetle tries to burrow into the bark of a tree, the tree's immune response is to undergo pitch streaming. So it puts out pitch, which is a highly flammable basically a poison, like a sap, but more concentrated and poisonous to bark beetles. And it uses that to push the bark beetles out through the holes that they're trying to drill into the tree. And in a healthy tree, when a bark beetle tries to burrow in, usually that pitching response happens pretty immediately. It immediately pushes it out and kills the bark beetle. However, when we introduce a stressor, be it a biotic or abiotic stressor, the tree needs to devote a lot more resources just to maintaining life. And unfortunately, the immune response takes kind of a back seat. So when the tree is stressed, it can't mount that proper pitching immune system response. And so it needs to devote more energy to other things just because of those other stressors. And that presents the perfect opportunity for Douglas fir bark beetles to come in, because once its immune system is down, they can really get in there and get into the phloem without having to worry about being forced out by pitch. And of course, the presence of bark beetles further stresses the trees out, which makes them an even better host for more Douglas fir bark beetles. And so once the bark beetles get in there, it's really the beginning of the end for the Douglas Fir. And after a thorough infestation, eventually the trees affected will die. And at that point, reproduction by the bark beetles is done, and since the bark beetles can't survive in a dead tree, that's kind of the end of the cycle for that specific stand for Douglas fir bark beetles. And this is kind of what that looks like from above, just a patch of dead trees in a circle. Not necessarily. This is more if it's a root rot infection, so it's like a localized thing, and then the bark beetles will come in and finish the job and kill the trees. If it's something like drought damage, that's more of spread out. So you'll see little pockets of dead trees here and there, not as localized as this. So to summarize, the Douglas fir bark beetle affects primarily stressed trees. Stressed trees are more susceptible, especially those affected by root rot. That's Phelinus weiryi for Douglas fir, although armillaria also affects Douglas fir as well as other species. Also trees affected by drought. So in large drought events, trees are also stressed. They're undergoing a lot of physiological stress, just maintaining moving photosynthate up and down. That presents a good opportunity for the bark beetles to come in. Also extreme heat, as well as ice events, ice storms, and after windfall events. This includes trees that have been thrown onto the ground but are still living. So when a Douglas fir is thrown onto the ground, it doesn't necessarily die immediately. It still is actually alive for quite a few months. And so those trees on the ground are a great host for Douglas fir bark beetles, at least until the tree dies. But also trees that have been affected by a windfall event that are still standing. So trees that are still standing but have kind of more internal structural damage. These trees are also good hosts for Douglas fir bark beetles. And also uniformly aged stands. So in stands that have been previously clear-cut, all the trees that grow up tend to be usually Douglas fir and more dense. And so these stands where all the trees are the same age and have low diversity, where it's mostly dominant Douglas fir, these stands are particularly susceptible to Douglas fir bark beetle infestation, assuming there's an introduction of another disturbance agent. So to measure Douglas fir bark beetle activity, I use something called the Lindgren funnel trap. That's what's hanging on the bottom right. So I use these to estimate beetle population and distribution by spreading them across my study areas. So the trap uses a three-part pheromone lure, the first part of which is ethanol. And ethanol in a stressed Douglas fir is released when the tree's endophytes are, when the tree is stressed, the endophytes can't get the energy they need, so they start undergoing anaerobic respiration. And so the second part of this pheromone lure is a pseudanol and frontalin chemical combination, and these are both Douglas fir bark beetle pheromones, one of which is released by female Douglas fir bark beetles within the trees normally, and that attracts males to come in and mate. And the other one is a general bark beetle aggregate. And this is released when there's large numbers of Douglas fir bark beetles together in a tree, and it signifies to other Douglas fir bark beetles that this is a good host, and it attracts more of them, which further stresses the tree and makes it even more of a good host. And the final part of this pheromone lure is a Douglas fir chiromone. And a chiromone is kind of like a chemical signifier for a species of tree. And so I use a Douglas fir chiromone to make my trap kind of, to a Douglas fir bark beetle, look like it's a Douglas fir. So together, the ethanol makes it look like it's a stressed tree. Pseudanol and frontalin make it look like it's being infested currently by Douglas fir bark beetles. And the chiromone makes it look like, or confirms to the Douglas fir bark beetles that it is in fact a Douglas fir tree. And the trap has all these cones stitched together, hanging like that, because each insect species, or rather family of insect species, reacts differently when they encounter an obstacle. I think it applies to most beetles in general. When they encounter an obstacle, they go down for some reason or another. And so cones work really well. So when beetles hit this, when they're attracted to this fake stressed Douglas fir, they fly down and immediately get caught in the trap, and they'll fall down all of these cones and eventually land in a little cup at the bottom that is just outside of this picture. But it's filled with a non-toxic antifreeze, which kind of traps the beetles, and then they'll sink. And then I'll empty it once a week and collect it and then count them. So my study for this part takes place in 3 different areas in the region. Furthest to the north is Lee Memorial Forest. This is a public access forest owned by the University of Washington. It's about half a square kilometer, a little bit more. outside Bothell. And as I said, it's public access, and it's actually surrounded by kind of suburban exurb areas. So this is kind of my most urban area. And I have 3 pairs of traps there. And just southeast of Seattle is the Cedar River Municipal Watershed. That's 368 square kilometers. And this, of course, is the source of Seattle's drinking water, or at least the primary source of much of Seattle's drinking water. And this is where a lot of my historical data comes from. Actually, almost all of my historical data comes from the Cedar River Municipal Watershed. And this year I have 9 pairs of traps. I had to lower it from 12 the previous year just due to access of roads that are covered by snow for the past few months. I just haven't been able to get up there. Last year I had 12 pairs of traps, and then going back in my historical data back to 2005, there's varying levels of traps based on availability of the employees at the Cedar River Municipal Watershed. And then, of course, Pack Forest furthest to the south. That's about 17 square kilometers. And this is a public access forest owned by the University of Washington. And my first year, 2015, working there, I had 12 pairs of traps. But it's such a small area, it didn't really add much to have so many traps, so I reduced it to 6 pairs of traps. And then this upcoming year, I've reduced it to 5 just because of ongoing experimental logging going on. At Pack Forest, I do a lot of experimental logging and different forage management practices, as well as different kinds of soil type studies. So this is the log transformation of my data from the Cedar River watershed of all years. So this is the counts that have been log transformed. As you can see, the counts, they vary quite a lot year to year, as well as within years. And so just looking at this, you can't really get a lot out of this data in this form. It obviously varies a lot, but there's a need to isolate it with temperature before we can really ascertain any kind of patterns coming out of this. So the connection with temperature, when we overlay temperature, becomes pretty obvious. As temperature goes up, beetles go up, and as temperature goes down, beetles go down. And this relationship looks like it changes as the year goes on, however. So at the beginning of the year, we can see it looks like a very strong relationship, but somewhere between day 140 and 180, it becomes a little less strong as beetles start to decline towards the end of the year, but temperatures still fluctuate up and down. So I wanted to look at or figure out why this change happens and when it happens, and if there's temporal subpopulations that have a different relationship with temperature. So are there beetles in the beginning of the year that have a different relationship with temperature than those emerging at the end of the year? And are those different temporal subpopulations, especially since it takes multiple months for the beetles to mature within the trees? So again, where's the divider between the early and late subpopulations, and what is the difference between these 2 temporal populations? So to investigate that a little bit, I correlated, this is the correlation with temperature of the log transformation of the beetle counts. And as you can see, the correlation with temperature decreases pretty steadily over time. It's almost linear, but there is an obvious drop in correlation at one point, and that is at day 157. At this point, correlation drops by 0.1, which is a fair amount compared to all of the other drops, especially because this is all of my data from all years and all sites, and it was consistently a drop at that day. So I'm calling before day 157 as early, the early flight, and after day 157, I'm calling the late flight. So I wanted to find out how significantly different are these 2 populations, and are they really differently correlated to temperature? It turns out they are. They're actually very differently correlated with temperature. The early flight was highly correlated. That's a correlation coefficient of 0.69, almost 0.7, which is, for ecological data, that's fabulous. That's really good data. Very strong relationship with temperature. Whereas in the second half of the year, that late flight is essentially no correlation, 0.032. So basically we see that there is, at least in some aspect, kind of 2 separate temporal subpopulations within the same year that have different relationships with temperature. So this is the cumulative proportion of all sites in years. And as you can see, not all historical sampling from the Cedar River Municipal Watershed, they didn't all begin before beetle emergence began. So especially in previous years, they had been putting the traps out after beetles were already flying. So they missed the early flight, and that's why I needed to continue getting more data and make sure I got my traps out earlier and earlier in the year to make sure I got those early flights, which are very important for determining that temperature relationship for when they start flying. But when you throw all of this together into one data set, it actually comes out pretty well, even with the missing parts of the data. So looking at the trend as a whole, it reveals that beetle emergence does tend to plateau around day 157 as well. So at this point, beetle emergence is nearing completion, and the number of active beetles are starting to drop. This varies year to year, of course, but that's when it begins, is day 157 across all of these 11 so odd years of data collection. So of course, I still have one more year of data collection to work on for this, and then more data crunching to work on. But going forwards, I plan on using Atkins's 1967 temperature rearing study, in which he demonstrated that the bark beetles could be raised to eliminate the obligate univoltinism. So they could reproduce more than once within the same year under ideal temperature conditions, which suggests that these are not obligate univoltinism. So using that, I aim to create a regional phenology model to identify where the flight temperature threshold is, so above what temperature these beetles begin flying in the beginning of the year. And on top of that, is it a flight temperature threshold? So do they immediately start flying when it reaches a certain temperature? Or is it an accumulation model? So does it have to hit that certain temperature for a certain number of days or weeks before they start flying? And once I begin crunching all this data after this upcoming year's data collection year, I should be able to get a good idea of that. And on top of that, I'll be able to determine the flight temperature inflection point. So that's probably around day 157, but I need to find what temperature happens at day 157 when beetles stop increasing, basically, and when that plateau is, when the beetle numbers that are active, when they stop increasing. And forward from there, I aim to create a predictive model to forecast when each brood's offspring will emerge. So those beetles that emerge in the beginning of the year, they're emerging as adults because bark beetles can only overwinter as larvae or adults. They can't emerge as, or they can't overwinter as pupa. So those first two emerge are those that overwintered as adults, and they're, of course, the first ones that are able to reproduce. And so I'd like to determine when their offspring are emerging. Is it in the same year, or is it in the following year? And it's believed that since they're the first ones that are able to reproduce and lay their eggs, it's their offspring that are the first to emerge in the beginning of the following spring, since they're already adults by that point. And on top of that, I would like to project how climate change-driven temperature changes, so an increase of 1.7 degrees Celsius, how that will affect the timing and number of emerging broods. So this is this past year's 2016 data with temperature overlaid. And so how will climate change affect the timing of emergence? So I predict that there will be an expansion of the active season with a lengthening of the favorable temperature conditions, especially as spring temperatures begin earlier. So we'll start to see that first emergence of that first temporal subpopulation brood emerging earlier in the year as spring basically begins earlier, like this. So as temperatures go up by 1.7 degrees and that typical spring temperature threshold moves earlier, we start to see the beetles emerging much earlier. So favorable conditions earlier in the year, beetles can come out earlier. This could mean that with an expanded length of time for the active season of these bark beetles, that the offspring of that first emerging subpopulation that has a greater correlation with temperature may be able to emerge at the end of that summer rather than the beginning of the following spring. So obviously very much lengthening the amount of time that these beetles are active and killing Douglas fir. Excuse me. So this obviously could change Douglas fir mortality dynamics and host immune response timing. And there's a need for a model like this in order to inform managers of how to alter management plans under changing climatic conditions, because this could very much affect how managers how they take control of their trees, especially considering when there'll be a lot more disturbance agents coming in, especially drought. We could be seeing not only more stressed trees, but a greater length of time for bark beetles to kill those stressed trees. So again, coming out at the end of summer. So moving on to my second objective, this is a little less connected to Douglas fir bark beetles, although they do play a small role in this. For this part of my research, I'm looking to quantify interstand variation in Douglas fir mortality and determine its relationship to a suite of potential explanatory variables, such as Douglas fir bark beetle densities, of course, as well as presence of root rot pathogens, relative stand age, stand density, and the proximity to other mortality areas. So I know you guys have had previous presenters talk about root rot pathogens, so I'll just go over the basics as it relates to this project. So for Douglas Fir stands, the primary root rot species affecting these are Pheromones and Armillaria. Pheromones, of course, affects primarily Douglas Fir, and Armillaria affects a wide variety. So stressed stands are more susceptible. However, they're not required. The stand is not required to be stressed in order to be infected by root rot, unlike the Douglas Fir bark beetle. It's considered to be a precursor to a Douglas Fir bark beetle infestation. So oftentimes root rot is that disturbance agent that comes in that allows bark beetles to come in after and finish the job. They are extremely persistent. They can live for thousands of years. Each individual root rot can live for thousands of years. And as such, they're often considered kind of a permanent part of the landscape. They're very persistent because of that. And they're a strong successional driver. They're often a major part of what drives the changes between different successional stages in a Douglas Fir forest to a more mixed species forest. So they form new genets genets are the individual root rots. They form new genets through mating of haploid spores, but in fact, most growth is actually vegetative, so just by growing, an individual growing. And they grow up to about 1 meter per year, and it does require direct root contact, or at least close to direct root contact. So obviously, old growth stands, spread is obviously much slower because the trees and the roots are further apart. And it spreads much quicker through younger, dense stands. And it kills, at least with Pheromones, it kills most, if not all, Douglas Fir in the stand. And as it's expanding outward, roughly in all directions, roughly in a circle, but that varies, which I'll get to, the center, as it's expanding out, the center of the infection area is being replaced by more resilient species. Especially in the case of Pheromones, it's being replaced by hemlock or cedar, species that are resistant to Pheromones, as mortality increases outwards and infects more Douglas Fir. And young Douglas Fir take about 1 to 3 years to die. I've not been able to find data for how that affects older trees, how long it takes to infect older trees. But for younger Douglas Fir, less than 80 years, it takes about 1 to 3 years to kill a tree, including the amount of time for bark beetles to come in and kind of finish the job. So as I said, it expands in a roughly circular pattern. It's not a perfect circle. It definitely varies based on local climatic conditions, local other conditions, moisture and such, and soil conditions. And so an infection of root rot can bring in, or frequently brings in, bark beetles. And so this can trigger a larger bark beetle infestation, of course, within the mortality area being affected by root rot, but also outside or in the trees adjacent to a mortality area, as well as trees outside of that. So beetles can kind of hit a crucial or a critical mass within the mortality area, and then on the outside of that mortality area, trees that aren't affected by root rot, they can get strong of a presence enough that they can overwhelm and overcome the immune response of even healthy trees. And this only really happens in more extreme situations. That's not entirely common. But in outbreak years, which you may have heard from about mountain pine beetle, this can happen quite frequently under those specific circumstances. So for some background on the Cedar River Municipal Watershed, this is where my study takes place entirely for this part of research. So the Cedar River Watershed was purchased from different logging companies between 1900 and the 1960s. And the dam was created in the early 1900s to provide the drinking water for what is now 1.4 million people, along with the Tult watershed. And so obviously there's a need to manage for water quality, including reducing erosion risks, obviously, which can cause a cascade of a whole other host of water quality changes. So as part of this management scheme, the employees at the Cedar River Watershed working for Seattle Public Utilities have a number of permanent sampling plots. They call them PSPs. These are the black dots on this map that they check every 5 years. And at each of these sites, they go and they create they lay out a 1/5 acre plot, and they're checking for DPH and density, and they're taking stand inventory at all of these sites across the entire watershed to get a good idea of what the conditions are around the municipal watershed and what's filtering everything going into the drinking water. And so overlaid on top of that, the orange areas, this is the bugs and crud layer from DNR. This is from 2015, since this data was collected while that data was available in 2016. And so I needed to make sure I wanted to collect data that kind of covered the breadth of the mortality gradient from high to low mortality to get characteristics of sand affected by different levels of ongoing mortality. So I needed to subset these PSPs by mortality level before going out, so I made sure I had a good representative sample across the entire mortality gradient. Especially, I needed a subset because I did not have time to go to every single one of these PSPs in the span of just a 2.5-year master's. So so high mortality level, I classified initially as those PSPs that were less than 150 meters from a mortality area, those orange striped areas. Medium mortality PSPs I identified as those within 150 meters to 1,000 meters of a mortality area. And the low mortality were those greater than 1,000 meters from a mortality area. And all of these needed to be within 500 meters of roads for ease of access and to save time, especially because I had interns and equipment that I was carrying with me. So the actual mortality of these sites I knew was going to vary, but I needed to make sure I got a full coverage of that gradient. And the bugs and crud layer from the aerial surveys from DNR was not entirely accurate, but it was decently close enough to get a good representative host of data. So at each of these PSPs, what the city does is they plot out a 1/5 acre plot and collect their data. I needed to make sure that I had greater spatial coverage through what are uneven mortality areas, like the picture on the left. These areas are uneven, and they're also not necessarily centered on the PSPs that currently exist. So I needed to increase my spatial coverage. So to do that, I first reduced the size of the center plot to 1/10 of an acre, but I also added 1/10 acre plots in the north direction, east, south, and west. And each 1/10 acre plot is 50 meters from the other, except for an addition of 16.1 meters from the center point to accommodate for the 1/5 acre plot that was already laid out by the city. And so in each of these subplots, I was collecting a variety of forest metrics, some of the usual ones like DBH for diameter at breast height for all stems above 5 centimeters, crown class, just an estimate for dominant, codominant, etc. I was also looking for the presence of Douglas Fir bark beetle Fras. That's the picture in the center on the bottom. It's like an orange sawdust almost. It's left over by Douglas Fir bark beetles as they're burrowing into these trees. And it's actually a mix of basically sawdust as well as Douglas Fir bark beetle poop. And it's a really good indicator of an ongoing, like currently ongoing infestation within a tree. I was also looking for signs of pitch streaming. It's the picture just to the right of the Fras picture. And like I said earlier, that's the immune response of the Douglas fir to something trying to burrow into it. This is a very good sign of an immune response to something like Douglas fir bark beetle burrowing into the tree. Not necessarily always Douglas fir bark beetle, but it is a good indicator of an immune response. I was also looking for the presence of epicormic branching. That's adventitious branching after a tree falls down in a stand and it creates a patch of light. And once it hits the side of, say, a Douglas fir actually notices that, and it will put out more branches in that location to take advantage of that light. And that's an indicator of maturing stand. And so within each stand, within each subplot, 1/10 acre subplot, I was also on the 3 most dominant living and up to 3 most dominant dead Douglas fir. I was looking for other mortality agents, such as Cryptoporus vulvatus. That's the picture on the far left. This is a fungal associate of Douglas fir bark beetle, and the beetle carries the spores into the tree. They're carried in little pouches under the arms, and once the tree has been killed by the Douglas fir bark beetle, the fungus kind of takes over and grows and starts breaking down the tree, and the little mycelial heads pop up from the little holes left by the bark beetle. And I was also looking for the presence or absence of root wrap mycelium within a foot above the soil. I was looking for within a 6 by 6 inch hole that I had cut into the bark of the tree, just the presence of that mycelium, which is like a white, almost latexy sign mycelium. That's pretty indicative of armillaria. So these are the results that I collected just from this past summer. And basically what you can get from this is everything varies, but it takes a lot more sorting to get some meaningful conclusions from this, which I'll go into. First of all, the sites look different individually. This is a high mortality site, or what ended up being a high mortality site. We get a lot more light coming through as there's a lot more canopy thinning, a lot more needle loss in here. A medium mortality site, these ones varied a lot. Some of them had a lot of light coming through, some had very little light coming through. And a low mortality site, very little light coming through. these are often more mature stands as well. So this is my post-sampling mortality level verification resubsetting. After I'd collected my data, needed to verify that high mortality sites that I'd previously identified, which ones were actually high mortality, because it did vary. So I first set that a high mortality site was going to be a site that had above 30% mortality, and medium mortality sites were those that had between 30 and 15% mortality, and low mortality sites were those with less than 15% mortality. And the spread was pretty good, and these three classifications were statistically significant. That's a p-value. These were all statistically significant, very significant from each other. This is the proportion of Douglas fir to all species across each mortality level. And this was fairly significant, at least between high and low mortality. High mortality had the greatest proportion of Douglas fir, and low had the lowest. Low also had the greatest variance. And medium and low had the lowest difference between these two. That remains to be seen why. I'll probably find out more this upcoming year. This is the proportion of Douglas fir with Fras. And this was actually not particularly significant, which is not entirely surprising, because Douglas fir bark beetle Fras is ephemeral. It doesn't always stick around, of course, because it's just the dust left behind from these beetles. It's easily removed by rain or wind. So high and medium did have more than low, but it was, as you can see from those p-values, not particularly significant, which isn't surprising. Now what really became interesting is this is the mean proportion of Douglas fir that had root wrap mycelium above the soil line. So for living trees, those are the bars that are not filled in, was between the living trees and the mortality levels was pretty inconclusive, and that's likely because the mycelium takes time to reach above the soil line. It starts in the soil and the roots. And so by the time it reaches above the soil line, oftentimes the tree is dead by then, so it would fall into the other category. With dead trees, of course, this was much more significant, especially between high and low mortality. That was very significant at the 0.004 level. Of course, high mortality, almost all of the dead trees always had root rot mycelium. In the low mortality sites, it varied, but in general, there was not very much root rot in these low mortality sites among dead trees. Medium mortality sites varied a lot. That's a very high standard deviation. And that's likely because many of these trees were probably in varying degrees of the leading edge of a root rot infection. So in some of these stands, the trees are just beginning to die. In some of these stands, the trees are already dying, varying degrees of infection going on in these medium mortality stands. And I think that's where most of the interesting data that I'm going to get out of this will come from, in those leading edge of the infection stands. So for canopy thinning, obviously this was very significant because it's a very good indicator of physiological stress. Obviously more thinning is more stress and more mortality. Medium mortality, again, is very telling because disturbance is just starting to increase in these. So in some of these stands, we're getting a lot of canopy thinning, and some of them not very much at all. And just because in some of these stands we're getting some infection and some not. And again, it's probably just like a not complete infection area compared to the high mortality areas. And these were actually fairly significant at the 0.1 to 0.01 level. So again, I still have another year of field work to go on from here. So this upcoming summer, I plan to sample for subsurface root rot mycelium at all of these sites. So I'm going to be digging into the ground to the roots and seeing if I can find root rot mycelium on the roots to get a better idea, especially in the living trees, which trees have root rot mycelium present in the high, medium, and low mortality zones. And I'm going to be checking the status of those dominant living and dead Douglas fir for changes and signs of changes of Fras and thinning and pitching, etc., just to see what's changed in a year. And also I plan to determine the potential relationships between all of these variables, as well as including some abiotic relationships for these, so slope, aspect, and elevation, as well as the distance to other mortality areas. And from there, I'm going to take a list together and identify stand characteristics that are associated with biological mortality agents. So say a high mortality stand in general has a higher DBH or low or greater percentage of Douglas fir, or has this slope or aspect in general. And so from that, I can project for sites that don't yet have increased mortality, but may be at risk of biotic disturbance agents because they have characteristics that are similar with sites that have ongoing mortality zones. So that's all I have for you guys now. I'd like to thank those from the University of Washington that have helped me so much, including my interns, Jake Betson and Constance Lynn. That's Jake on the bottom right shimmying across a log in one of my sites, as well as fellow graduate students Sean Callahan and Riley Metz, who've helped me a couple times over the summer. My advisor, of course, Patrick Tobin, who's been my very helpful helper for learning how to do all of this, because I'm relatively new to forests. I studied in Florida for undergrad, so this is all pretty new for me. And he's been very helpful in helping me learn all of this. And then, of course, my helpers from the Seattle Public Utilities, especially Amy Labarge, who was pioneering the Douglas Fir bark beetle study before I came in, and who's always had lots of information to contribute, especially since she knows these forests at the Cedar River like the back of her hand. And Rolf Gersand, who has been pulling a lot of the strings behind the scenes to make sure everything goes smoothly. And of course, thanks to University of Washington and Seattle Public Utilities for their funding to make this study possible.